That brings us to chapter 12, where we are going to talk about lipid peroxidation. And I don't know if you remember before, but I sort of mentioned that lipid oxidation and lipid peroxidation are two terms that are used pretty much synonymously. And what we're going to talk about in this chapter is we're going to look at lipid peroxidation and discuss it a little bit from an aspect of biology in terms of structures that can become damaged in the skin in particular. And then we're going to also look at formulations and how lipid peroxidation is important when you're considering formulation strategies. So in the first section, we'll talk about lipid membrane peroxidation, and this is discussing how lipid peroxidation is important in an in vivo environment. We'll discuss a little bit UV-induced radicals in the epidermis, and essentially what are the skin lipids that are most susceptible to lipid peroxidation. Then we'll go on to discuss lipid peroxidation in bulk oils and colloidal systems. And then we'll briefly talk about the polar paradox, which is kind of an interesting phenomena that occurs with antioxidants, in particular with reference to formulation type systems. So in membrane lipid peroxidation, if you can imagine that this diagram is just half of a lipid membrane, so you would have the head groups, the polar head groups of the phospholipids are shown here on the top. Those are in contact with the water phase. Now that can be in the interior of the cell, so this could be like the cytosol, for example, or the cytoplasm. And then you would have the aliphatic chains going in the other direction, in the opposite direction. And then, of course, you can imagine that there would be another monolayer down here, which would form this overall bilayer. So we're just showing half of the bilayer for illustration. And the key points here to look at are we have a phospholipid, and when you have points of conjugation, as we do in this phospholipid shown here, this is what can be susceptible to lipid oxidation or lipid peroxidation. And not to get too far off target or off topic, but when we discuss natural ingredients that are used in a lot of the formulations, that actually requires a lot more care and understanding of oxidation, and you have to often use more antioxidants in your formula to compensate for that, and that's because natural materials tend to contain more double bond type structure. And let's get back on track here. We're talking about cells in the body or in the skin. This is the phospholipid membrane. We do have the double bond type structure, and as you can see here, this can interact with this free radical, and you end up with a delocalized electron in this where you had the double bond structure before. You're left with RH, so this radical becomes neutralized, and then the phospholipid that now has a radical tail on it interacts with oxygen to form this sort of peroxyl type species. So you have a radical still formed here. You can see the double bond character is sort of introduced back into the structure, but now this is changed from being a hydrophobic environment down here to actually becoming a more hydrophilic one. So as you can see in part C here, these two oxygens with the radical actually migrate up to the membrane surface, the interface with the water or the cytoplasm, and the rest of the tail remains in the lipid phase, but part of it is at the interface in contact with the aqueous environment. Here we show alpha tocopherol, and this is a demonstration on how alpha tocopherol would help to neutralize this. Alpha tocopherol has this long phytyl chain, if you recall from our previous chapters that we talked about alpha tocopherol structure, that keeps it embedded in the lipid membrane, but at the same time it has portions of it that are more polar in nature or more hydrophilic in nature, such as this hydroxyl group, which is in contact at the interface of the lipid membrane or the bilayer with the water structure up here. So as this is in contact with that, what we end up seeing is that this donates a proton, so the alpha tocopherol neutralizes this free radical species. And then you can see down here in part D that you have this HOO structure attached now. So it's neutralized at this point. Then you create a tocopherol radical species, as you can see here. So, you know, where the hydrogen was attached to the alpha tocopherol, now it's a free radical species. Remember we talked about ascorbic acid, or in this case ascorbate, actually regenerating alpha tocopherol, and it does that as this is at the interface, at the aqueous interface here. You can see that ascorbic acid, which is an aqueous soluble antioxidant, can simply come through here, donate a proton to this, and basically regenerate alpha tocopherol, which is shown over here. In doing that, ascorbic acid itself becomes spent as an antioxidant, but you know, as we mentioned before, this can be later regenerated by glutathione, the antioxidant glutathione as well. Okay, so what happens from this point is, remember, you still have this lipid, phospholipid, which has essentially a damaged chain, but fortunately we have various enzymes, and this is just showing you an illustration that these enzymes would sort of operate on or work on the phospholipid. This enzyme is phospholipase A2. This one here, GPX, is glutathione peroxidase. This FACOA is fatty acyl coenzyme A. And then, of course, we have this last one, which is phospholipid hydroperoxide glutathione peroxidase. But essentially, what I want to show here is just, you know, various antioxidants work on repairing the phospholipid and restoring it to its original structure. This is just a description of, this is for your reference. This is a description of the slide that we just showed on the previous slide. So then we can talk about UV-induced epidermal free radicals. So imagine starting with a lipid molecule, and we'll just call it LH. And the lipid molecule loses an electron. Now this occurs, one of the ways that this occurs is via this Fenton reaction that's shown down here. This is a very well-known reaction in chemistry. And what it consists of is that you have iron plus oxygen that is converted to Fe3 plus the hydroxyl radical, and the hydroxyl radical is what's really dangerous. The hydroxyl radical then interacts with the lipid LH, forming the lipid radical. And also note that, you know, before this even happens, the way this whole reaction sort of starts off is that you actually have Fe3, you have iron 3 plus in the cellular environment. You have molecular oxygen, which is via UV light converted to superoxide anion. Superoxide anion actually catalyzes the conversion of Fe3 plus to Fe2 plus. Fe2 plus reacts with hydrogen peroxide, and that produces the hydroxyl radical. So we're left with this lipid radical over here, which in the presence of oxygen results in this lipid peroxide radical, LOO dot, which is shown here. LOO dot can further react with other lipids, extracting a proton or a hydrogen atom from those, resulting in another lipid radical. And of course, this process would be like a chain reaction, and you would continue to damage more and more lipids as this process would continue. The more, another lipid radical would react with another oxygen molecule, giving you another lipid peroxide radical. And this is actually, this lipid peroxide radical is actually neutralized by the other lipid that it reacts with, producing LOOH. And then LOOH can further end up being broken down again to a radical species. It could be either a lipid alkoxyl, LO dot, or it could be a lipid peroxyl. This is actually misspelled. There shouldn't be an O before the Y in this the spelling here. So some of the unsaturated fatty acids, PUFA stands for polyunsaturated fatty acid. What it means, poly means you have many or several degrees of unsaturation, which unsaturation of course means double bonds in your fatty acid structure. And PUFAS or PUFAs are known, as I mentioned already, to undergo lipid peroxidation, basically due to this double bond type structure. These are several important ones. These are actually, in this I'm talking in general, or I'm speaking, generally speaking, about lipid peroxidation in the body. These fatty acids are usually incorporated into phospholipid structures. So the long chains that you have on phospholipids, they just don't come from anywhere. They come from fatty acids. Basically, the phospholipids incorporate the chains of these fatty acids into them. And these are two different types of important fatty acids, which you've probably heard about because everyone talks about them in terms of their health. These are the omega 3 and omega 6 fatty acids. And essentially what it means, omega 3 just means that when you have this conjugated fatty acid structure, if you count how many carbons is the last double bond from the end of the molecule, it's 1, 2, 3. So this is an omega 3, the alpha linolenic acid. And then these other three are actually omega 6, 1, 2, 3, 4, 5, 6. So gamma linolenic acid, arachidonic acid, and linoleic acid, these are all omega 6 fatty acids. So again, these are important molecules that they're good for you in terms of health, but they are targets of lipid peroxidation. In the skin, we have a number of things that can be affected by lipid peroxidation. And if we start at the lower layers of the epidermis, or even with some of the cellular components of the dermis, we know that at those layers, the cells consist of phospholipid membranes. And so you would have phospholipids such as, this is just an example, you have a number of different types of phospholipids, but here you can see this one has some double bond type structure to it. This of course would be susceptible to lipid peroxidation. We also have triglycerides that are present in skin, and these can be derived from palmitic, oleic, and linolenic acids. And you can see here that you have various double bond type structures within this triglyceride. So of course these can be susceptible to lipid peroxidation as well. And squalene is present in skin, and so is cholesterol. Squalene is actually a precursor for cholesterol. But both of these structures are possible targets of lipid peroxidation in the skin. And we find these not only in the viable epidermis, but also in the stratum corneum. Specific to the stratum corneum and to the stratum granulosum stratum corneum interface, the ceramides that are most susceptible to undergo lipid peroxidation are these three, and essentially they're the lipids that have the ester component here, and this last part, as you can see, is essentially a 1, 2, 3, 4, 5, 6, an omega-6 fatty acid in each case, and of course this portion of the molecule can undergo lipid peroxidation. So the other ceramides that we have that basically don't have these sort of conjugated systems in them, they're not going to be susceptible to lipid peroxidation, at least not readily. The double bond type structure is really what is susceptible, and usually when you have saturated type fatty acids, these tend to be more stable. So let's talk a little bit from a formulation standpoint, and how antioxidants are used in these colloidal type systems. And we'll just talk about bulk oil systems first, because bulk oil systems were studied very much in the food industry, as you can imagine, because of all the oils and things that are sold in supermarkets. And there's always these to monitor shelf life and to understand spoilage of the oils. A lot of research has gone into understanding how antioxidants could work in helping to preserve oil systems. But there's a lot of interesting things from this that we can sort of use in cosmetic science. And then of course oil and water emulsions. We could be talking about water and oil, or any type of an emulsion, but we'll just give you an example of this. And then also liposomes. So when we talk about bulk oils, imagine that you have a bulk oil in this vessel here, or this flask. So keep in mind that the bulk oil has a lipid-air interface. So the first question you would probably want to ask yourself is, which part of this is most likely to be oxidized? Would it be the bulk oil or the bulk lipid? Or would it be at the interface? And the answer is the interface is the most susceptible to oxidation, because you wouldn't really have dissolved oxygen in the oil phase. The oxygen would be above the oil phase at the interface here. And if we look at the emulsion systems, so this, that makes us think about something. Just to go back a second to the bulk oil system. If we were to choose an antioxidant to protect this, we wouldn't choose, since we know the oxidation mostly occurs at the interface, we would not choose an antioxidant that would be down here only in the oil phase, because there wouldn't really be anything for it to protect down here. So we would want to find an antioxidant that would migrate to this lipid-air interface and basically stay in this area, and that's where its activity could be. The same with emulsion systems. The water phase, the interface between the actual inside these oil droplets in the oil and water emulsion, deep inside of them, you really wouldn't have as much susceptibility to lipid oxidation. But at the interface of these lipid oil droplets with the water phase is essentially where you would have the most susceptibility to lipid peroxidation. We could also discuss liposomes, micelles, bilayer sheets, various types of colloidal systems. Just thinking of a liposome in general, liposome is essentially like a bilayer, but it's formed into a sphere. You can kind of see this cross section here, what it looks like. And we'll just to give you an idea, we'll make some mention to that in a few minutes. So, for this system, what's most likely to undergo oxidation? Well, mostly probably in this lipid phase here. So we want an antioxidant that's basically going to migrate in some way into this lipid phase, or it's going to stay in this lipid phase here. So some of the things that are known about using antioxidants in colloidal systems, one of the main things is called the polar paradox. And essentially what this says is that polar antioxidants are more efficacious in bulk oil systems than non-polar antioxidants. So again, that'll seem kind of weird to you, thinking, our natural instinct would be, if we have a bulk oil system, which is a non-polar bulk oil system, our natural instinct would be to use a non-polar antioxidant. But as I mentioned, the region most susceptible to lipid peroxidation is at the interface, and for that reason, actually polar antioxidants are more efficacious than non-polar antioxidants. And essentially that's because polar antioxidants will migrate to the air-oil interface when you have a bulk oil system in that. Okay. The same basically occurs in the emulsion systems. Something that's going to be at the interface again of the lipid and at the lipid water interface is going to provide more protection than something that's going to be strictly in the oil phase or strictly in the water phase. We also know that electrostatic interactions are really important in determining antioxidant efficacy, and that you can imagine that if an antioxidant has a charge, if it's an opposite charge or the same charge, it's going to either be attracted or repelled from another species. So, just as an example, it's been shown that charged antioxidants are more efficacious in liposomes than non-charged species. In addition to that, we should expect that the pH of the formulation will also influence the ability of a charged antioxidant to interact with other charged species, because as you change the pH, you could change the charge on the antioxidant. Okay. So, just as an example here of looking at electrostatic effects or charge-charge interactions, it's a lot of results have been conducted or shown that basically negatively charged antioxidants, for example, ascorbic acid and Trolox. Ascorbic acid is, as you saw already, this is a negatively charged antioxidant. Trolox is a water-soluble version of vitamin E. This is actually, it has a negative charge on it though. But it's seen that these are actually more efficacious in micelles of linoleic acid that are stabilized with a cationic emulsifier. The specific emulsifier is given here, hexadecyl trimethyl ammonium bromide. Then if you look at the same micellar system that's stabilized with an anionic emulsifier, such as sodium dodecyl sulfate, and essentially this occurs because you have repulsion between the negatively charged antioxidants and the negatively charged head groups of SDS, which keeps the antioxidant away from this sort of interfacial area to provide protection there. And with the cationic emulsifier, you basically have an attraction between ascorbic acid and Trolox. So that negative negative antioxidant with positive emulsifier sort of brings the antioxidant to the interface right in the area where the liposome is most susceptible to undergo lipid peroxidation. So that concludes this chapter, and you can find basically a full reading section on this in chapter 5 if you wish to sort of get a better overview of some of the topics we discussed in this class. So that will be it until next time. Thank you for your attention and enjoy your day.